Daniel is a biologist specializing in animal engineering and rodent breeding management. He leads the breeding coordination, scientific operation, and comparative medicine team at Novartis Biomedical Research, where he oversees the breeding activities for genetically engineered models and optimizes operational processes. And Filippo has studied archaeology and biology. I guess today we will talk about the biology part. Filippo serves both as Vorarbeiter, I don't know what in English, and Tierpfleger at the animal facility at Novartis Pharma in Basel, where he brings extensive experience in transgenic mouse breeding. So together they will present the first talk of today, entitled Breeding of Genetically Engineered Mouse Models, Challenges, Opportunities, and Lessons Learned. So please join me in welcoming Daniel and Filippo.
Hi, everyone. I hope you can hear us well. We're doing this on a small laptop, so we're going to come off video, but we're going to be back later for the questions. I'm sharing my screen now, and please let me know if you can see the presentation well. If somebody could confirm, that would be great.
Yes, we can see it.
Many thanks. All right, so my name is Daniel Breustedt, and here with me today is Filippo Gallegra. So we're basically covering the breeding coordination and then also the actual vivarium work, so setup of matings and so on and so forth, hoping that this is a nice mix of presentation of our work. The title, as mentioned, is Breeding of Genetically Engineered Mouse Models, Challenges, Opportunities, and Lessons Learned. And with that, I would say we dive right in. So basically today, there's going to be a very short introduction about genetically engineered models, just 1 or 2 slides. Then I'm going to talk about what we call phase 1, which is from managing space to managing colonies. I'll follow that up with a phase 2, where we started forecasting cage numbers. Then Filippo will give you the perspective from within the animal facilities, so the animal caretaker view, if you will, and there will be a very short summary. So when we talk about genetically engineered models, just very quickly so that we are aligned, this term we use for transgenic models, knock-in models, knock-out models, and what they all have in common, or really most of them have in common, is that they require genotyping. You can purchase some of these engineered models off the shelf from the known vendors, but the majority of these models are not available off the shelf, right? So they either come out of internal animal engineering efforts, or they have been generated at a third party like university, and we in-license them. And for that reason, we need to breed them ourselves. So at any given time in Basel at Novartis, we have something like 120 to 150 lines, active breeding. And since projects sometimes are discontinued and replaced by new projects, we have a turnover on average, I would say, of 30 new lines per year. So 30 lines disappear on average and are replaced by 30 new ones pretty much every year. The lines we breed carry single or multiple genetic modifications. And here in-house, so on the Novartis campus in Basel, we breed them in IVC racks, which you can see a picture of on the right-hand side. We have also outsourced part of our breeding to a CRO, and there the animals are bred in isolators. So as Torsten mentioned in the introduction, when you're breeding genetically engineered models, there's quite a few challenges, right? So the first challenge, I believe, is that the number of available gems and also their complexity is increasing continuously. So if you think back some 10 years, maybe a bit more, you would mostly have transgenic and knockout lines carrying single mutations. Every now and then you would have a combination of a Cree and a Floxed knockout allele or something like that. But it was really not as complex as it is now. Going over our current line inventory, we have 50% lines carrying 1 mutation. We have around 30% carrying two mutations, so that could be a Cree or Cree ERT2 in combination with a conditional knockout, so that you can induce the knockout. And we have around about 20% of mice or lines carrying 3 or more mutations. So you could expand the example I just gave and add maybe another conditional allele or a lock-stop-locks EGFP or any other reporter. So we have quite a few lines carrying 3 and 4 mutations. The maximum number we've seen is 7, but that was a one-off. But I think it's very clear that because these lines and all the traits inherit independently, breeding and also handling of the breeding is fairly complex. And one of the factors you see often is that you get a great number of mice with unneeded or undesired genotypes. So in short, managing these complex lines is really a challenge for every breeding co-facility. I would like to expand a little on what I mean regarding complex lines. So imagine you have 3 baselines depicted here in orange. One is an inducible reporter line. As I just said before, it could be a lock-stop-locks EGFP. The second one is a conditional floxed target allele, right? That's your gene of interest. And the third one is a CRE line. That could be CRE or CRE-ERT2. It could be tissue-specific. So there's many, many CRE lines out there. And the goal is to combine these 3 alleles into one line. And what you want to achieve really is that in a cell-specific manner, driven by the CRE, which should be cell-specific, you want to generate the knockout. So basically turn the floxed conditional knockout into the full knockout. And at the same time, you want to mark these cells with EGFP by excising the lock-stop-locks cassette and activating the GFP marker. Now, obviously, 3 alleles, bringing them into one line is not done in one breeding step because you can only ever breed 1 male and 1 female. So the approach would be that you first combine the conditional knockout and the reporter allele. And assuming that your baselines are homozygous, you will have 100% heterozygous, double heterozygous animals here, unless one is on the sex chromosome. So you would do the same thing in parallel, combining the conditional knockout and the CRE line. And again, you have 100% double heterozygous offspring coming out of this breeding. Now, if you want to combine the 3 alleles, the obvious next step is that you combine, that you breed these 2 intermediate lines to each other, and thereby you generate a line carrying the conditional knockout, the reporter, and the CRE. But it's not that easy, right? Because essentially, if you look at the breeding scheme, the conditional knockout is heterozygous in the intermediate lines. So essentially, this is a head-to-head mating, and we all know that out of this, you get 25% homozygous animals. At the same time, the reporter coming from the baseline or from the intermediate line is heterozygous, so it's inherited with a 50% probability, meaning that out of your 25% homozygous animals down here, only half, so 12.5%, will also carry the reporter. And the same thing is true for the CRE. It's also inherited with a 50% probability. So out of your homozygous knockout or conditional knockout, carrying the reporter, only half, and that's 6.25%, are going to carry the CRE allele as well. So in other words, if you follow this breeding scheme, you will get 6 animals with the desired genotype, and you have to breed 100 animals. And that's only assuming that the Mendelian inheritance works as it should, which may or may not be the case. And that's already quite a bad percentage of useful animals, and it gets worse because if you consider to breed 100 animals of your experimental line, and assuming that you have an average litter size of 5 pups per female, you would need to put 20 females of these intermediate lines into mating. And when you have 20 females, you have also generated 20 males. So you have bred 40 animals for the intermediate lines. You have bred 100 animals for your experimental line. That totals up to 140. And you get 6 animals out of there that you could use for an experiment. So that's not a very sensible approach. I think we all agree to that. Much more rational would be to try and bring at least the conditional knockout in the intermediate lines to homozygosity, right? So we can see later the reporter ideally is also homozygous. The CRE could be heterozygous because sometimes you need controls without CRE, and then you can get these out of that. But at least if you have the conditional knockout homozygous, then all the offspring in the experimental line will be homozygous already for that, right? And what you would need to do is you would need to breed the double heterozygous animals that you received in the very beginning back to the homozygous line here. So you add one generation, but then essentially, if you bring this or if you implement this breeding scheme, you will have 50% animals carrying the genotype hom het, which are your experimental animals, and another 50% which be hom het wild type and can serve as controls. So that's a lot better regarding ratios of useful animals. And as I said, I mean, we have lines carrying 4 alleles. So if you add, for example, another conditional allele to this mix here, it gets even more complicated, and setting up good baselines or good intermediate lines makes even more sense. With that, I hope we all have a better understanding of what we mean in this talk by complex lines. And I would like to move on to our situation in Basel in 2015. So back then, most or a lot of our mating was really based on managing space. And that led to quite a few challenges. So we usually had a very high occupancy in the breeding facility of 90+%. So our total capacity, 45 racks, 3,645 cage spots. And that means we usually had something like 2,800 to 3,200 cages in there, which is really, really high occupancy. The problem with this is that when you're breeding, you need to wean animals on a regular basis. And so basically, often you don't have space. You need to remove surplus animals in order to create space. And that's a reactive approach where you rely on people getting back to you if they actually need the animals that you would like to eliminate, and not really a good situation. Another problem was that the lines were distributed over several racks, which is obviously problematic for hygiene monitoring if you have a sentinel-based or rack-based hygiene monitoring regimen. And then obviously we bred back then very many animals, which is not good regarding the 3R. And it also leads to a very high workload downstream if you think about genotyping, but also cage washing and all the other factors. So to summarize, back then we had limited filtering and alignment of the breeding goals. So the situation, how we handled and how we managed the breeding back then looked, at least for one research department, like this, and I'm going to come back to the others in a second. On the left-hand side, you have the researcher with the breeding request. And for one of our departments, which was the one doing the animal engineering back then, they literally transferred their breeding requests directly to the animal caretaker, right? So they basically had a list of things they wanted the animal caretaker to implement. They sent that list, and the breeding team had to do that. And only, as I said, when, for example, there was problems with space, the animal caretakers would feed back to the researcher and try to solve the problem. What we did have already, which is not the focus of today, is a genotyping lab. So quick turnaround times for the genotypes of around about a week. That's helpful already because it drives decision making. And we have an embryology lab that allows you to freeze down lines. But still, in the core business here, in the middle, in the breeding, this process with this department was really not ideal. For other departments, we had what we call breeding coordination, so we could provide expert input from experienced animal caretakers. They would talk to the researchers to find out what they want. And we really decided to take that approach and to strengthen it a lot. So what we did is we implemented a strong breeding coordination team with 3 experienced animal caretakers that also have project management skills. And this team of 3 basically meets on a weekly basis. We discuss the lines. We discuss, for example, also with the animal caretakers space and so on and so forth. So the new approach was really for the researcher and the breeding coordination team to meet when a new line, for example, is brought in or when a new breeding goal needs to be discussed. And then you agree on breeding goals. The breeding coordination team does all the calculations in the back. It communicates with the animal caretakers, implements the matings, starts them, and more importantly, stops them at the right time. The animal caretakers always have somebody that they can reach out to. So rather than having a researcher that might be on vacation or just too busy at the moment, the breeding coordination team is there and can be reached. I mean, usually within half a day maximum, they would get an answer. So this is the approach we took, that we basically developed out of the one before. And with that, I would like to show you what we implemented and what we achieved doing this approach. Very quickly, I'm going to bother you with a definition slide. You don't have to go through all of this. All I want you to know is that the animal numbers, weaned animals and delivered animals, we have very accurate data because we track that in our respective breeding and ordering databases. With this, let's take a look at the data. So what you see here is monthly animal numbers from 2015 to 2017, weaned versus ordered. On the x-axis, you have time, so that's the years. On the y-axis, you have animal numbers. And the blue curve represents the number of animals weaned, again, from our breeding database. The red curve represents the number of animals ordered from our breeding into experiments, and that's exported from another database. And what you can nicely see that over this 3-year time, the number of weaned animals is continuously reduced, whereas the number of ordered animals that we delivered into research remains more or less constant. I mean, you can see the usual ups and downs month over month, but I think the trend is quite clear. So for full clarity, in 2017, we also decided to outsource part of our breeding to a third party. That decision was mainly driven by space because we had quite a bit of refurbishment going on here on campus. The goal was to move roughly 50% of the lines offsite, and we decided to do that mostly for the more simple lines. Animals ordered from that third party, you can see here the green curve, and you will realize that this only picks up in the middle of 2017. So that was certainly not a factor in this reduction of animal numbers that you're seeing here. If we look at the yearly numbers, so it's again the same data, only summarized per year. Regarding animals born or weaned, we came from 36,000 in 2015 to 29,000 in 2016 and 23,000 in 2017. So we'll see that there's a delta of 7,000 animals between 15 and 16, and there's a delta of 13,000 animals between 15 and 17. So overall, you could say that we saved around 20,000 animals over 2 years because we essentially didn't breed them because we managed the breedings the way we did. The ordering ratio, that was the red curve. You can see here, it remained more or less constant, right? So looking at the 2017 number, that's our internal deliveries only, and there's an additional maybe 400 or 500 animals that came from the third party. That was the green curve that picked up towards the right-hand side of the graph I showed on the last slide. And then if you calculate the ratio or the ordering percentage, you can nicely see that it went from 23 to 28 and up to 35%. So that's a very significant increase. I'm also happy to say that for this work, we received a Novartis Internal 3R award at some point. So it was recognized within the company, which is always nice. But it also had plenty other consequences. Because if we look at the cage numbers in breeding, and this is the total cage numbers, so this could be split between internally and externally, but it's not really the point here. The point is that in 2015, we had above 3,000 cages on average in our breeding facility. And we were able to bring that number down by 2018 to around about 2,000 cages, and we stayed on that level. So that's roughly a 30% reduction in cage numbers, and obviously also in footprint, in cage washing, and so on and so forth. Since 2018, we also have month-over-month data. This is just as an inset here at the bottom, and this is to illustrate that this is not going up and down crazy from month to month, and it's just the yearly averages that are the same. I mean, you can basically draw a line there. We are able to maintain our cage count at around about 2,000. So I showed you that we were able to reduce the numbers and that the output was the same. And I'm now showing you the output for the whole period till 2022. What you see here, again, on the x-axis is time. On the y-axis, it's the number of delivered animals on a yearly basis. The blue curve is the internal one. And remember, in 2017, we started outsourcing. So naturally, that curve came down. The orange curve is the number of animals coming from the external breeding. Obviously, that's picking up. And the gray curve up there is the overall numbers. And you can nicely see that in spite of a reduction of cages from 3,000 to 2,000, our output has remained fairly constant over the years. What I'm not showing you for the whole period, I showed that for 2015 to 2017, the number of animals bred and genotyped was reduced in parallel, and we were able to maintain that on that lower level, and that points at an increased efficiency. Now, obviously, in a year like 2020, where COVID hit, that ratio was a bit lower because everything came to a halt and then had to be restarted. But overall, I think it's quite clear that we were able to increase our efficiency quite a bit. Now, with this done, basically just as a high-level information, how did we achieve that? I mean, one thing is the breeding coordination team really spent a lot of time optimizing breedings, understanding breedings. And one thing we did right away is to not only talk to the individual researchers, because if you talk to a researcher, very often you will hear that the line is extremely important and that it cannot be stopped, it cannot be cryopreserved, it cannot even be reduced in breeding. But when you talk to the whole research department, it may come up that certain projects have been discontinued and the line is around because the researcher has some interest in it, but it's not necessarily a project that's strongly supported any longer. So in these meetings, we were able to shut down quite a few lines, and that was extremely helpful. Then obviously, with the breeding coordination team in place, we have very regular and active communication. In a way, it's a collaborative approach between the researchers and the breeding coordination team, but definitely the breeding coordination team taking ownership of the breeding approach. Another factor is really also that we have a lot of communication and alignment within our own team, right? So typical case is that the researchers want to start experiments when they come back from the Christmas break in January. And in the past, they would all set up their matings at the same time, leading to a massive peak in breeding. And now with the breeding coordination team, we are really able to shift and to balance these peak seasons also after the summer break. On top of that, the breeding coordinators can learn from each other because very often you have a problem that somebody else has had in the past and there might be a solution already. And then obviously a lot of work also goes into the more stringent monitoring of lines. So while a researcher will always be happy to set up a mating to generate these animals, he may not be so good at stopping these matings, and you may get 2 or 3 or more litters, whereas 1 would be enough. So this is something the team monitors very closely. So with this, with the here and now under control, another thing, and that was around about 2018, another thing we realized is that we're still pretty much blind for what's coming in the future, right? So we never knew, is there going to be an increase on the horizon or a decrease? So we tried to come up with a solution for that, and we thought about many complex things like extracting data from a breeding database, but in the end, we decided to try something very simple. We said we just define 4 simple line categories: maintenance line, small production, medium, and large production. And for all of these lines, we define an average cage number. Like maintenance would be 10 cages on average over the year. Now, obviously, when you're about to wean the new generation, that's going to be more, but then it's going to be less in an intermediate phase. And the same for small, medium, and large production with 50 and 30 and 60 cages respectively. And only if something is really, really big as a project, we might put in a different number. So we have a list of all of our projects. Every line in there gets one of these numbers associated. And then in Q4, we can do a forecast for the following year. And what I'm going to show you now is for one department, the forecasting versus the actual numbers. So on the x-axis, you have the different years. On the y-axis, you have the cage numbers. And then the blue bars are always the forecast in Q4 of the previous year. and in orange are the actual numbers, yearly average for the year that you forecasted. So what you can see for 2019, we forecasted with the blue bar close to 500 cages for this department. We ended up having an average for the year of around about 400. Then we forecasted an increase to 500, maybe 40, and we did see an increase from 400 to 460. That's the orange bar. Then for 2021, there was a bigger increase in the forecast beyond 700. And the actual numbers also came up. And then in 22 and 23, the forecast and the actual numbers went down again. So you can see two things here. First of all, obviously the forecast is far from being perfect, right? And that makes sense because if you say, for example, you sit and you meet in September or October and you go through all the lines, you come up with a certain number, that's the blue bar. But then if certain lines get discontinued over the course of the year, you may end up with a number that's not 100% accurate. But it definitely shows you trends, right? So if you see, for example, from 2019, under 500 to 2021, close to 700 cages, this is something you would like to know in advance. And with the forecasting, we do know that. So that's a neat and useful tool, and it's not a lot of work. And with that, at this point, I would like to hand over to Filippo so that he can provide you with a perspective from the vivarium and what all of that actually means for him and his colleagues.
So, hello everyone. So what does this new breeding approach mean for animal caretakers? First, I would like to talk about my feeling of purpose. Why do I choose to work in an animal testing facility? It's because I believe animal testing to be an important part in drug development. At the same time, I have a compassion for the animals we use. So why does managing breeding by space diminish my feeling of purpose? For me, it's all about perception. Overproducing and prolonged housing of stock mice with no real need dilutes my feeling of purpose. Holding 30% more cages for the same number of mice delivered and euthanizing way more mice than you deliver just doesn't feel right for me. And then what about compassion toward the animals we use? We can and we should keep improving the care for our mice, but the lab environment will never be an ideal place for them. So, in fact, overproducing mice is not very desirable. So what does managing breeding do for us? It's much more engaging for me as an animal caretaker. I know what the goal is, and I know the purpose of each mouse is defined by the needs of the research. The information why any given breeding task is performed can be understood by also the animal caretaker. Put together, for me, the most satisfying breeding task is delivering mice into a study. It's not taking care of the mice, it's really delivering them in a study. I like to take care of them because that's very important, but sending them to a study is the goal. Anything else is just senseless. So it also reduces the workload for the animal caretaker and makes his work more fulfilling. Because overproduction and letting mice age for no real reason leads to more overall work for the animal caretaker, with the same outcome, the number of mice going into studies. Now we have 30% less cages and mice to tend to. And on top of that, if you're at an occupancy of 90%, you're going to be shifting a lot of cages around each week just to make space for the weaning. And think about euthanizing a lot of excess mice you never should have produced. That takes time and can lead to compassion fatigue. So what do you do with the saved time? Well, the saved time is well spent with dedicated coordinators doing the breeding planning and breeding staff conducting extra pregnancy and litter checks to then be able to decide if you are on track with your breeding plan or need to stop or start breeding as needed. In this whole thing, an important thing you should be aware of is the workload per cage is significantly increased. So you can't expect an animal caretaker to handle the same number of cages as before, because the reduction, those 30% we reduced, those are more or less long-time stock animals, and they are the less work-intensive cages in a breeding. So now I would like to also talk about misunderstanding, missed timelines, and unnecessary aging of mice, and then also clustering of breeding tasks. So let's look at the top depiction. In that setup, we had more than 30 researchers doing the breeding planning. So you end up with 30 plus individuals communicating their breeding tasks by email, by telephone, or Excel lists. And that can be a real nightmare to keep everything in mind. And I often had to then say, well, I forgot what was in the email 3 or 2 or 1 month ago. It's really, really, really difficult. And then for misunderstandings. For some, researcher stop the breeding could mean remove the male and wean all upcoming litters, or it could mean euthanize the entire breeding cage with all current litters. I think it's hard to align on a consistent terminology that reduces misunderstandings. In this setup on the top, the researchers have a good understanding what their requirements are. But should they be bothered to translate those into a breeding plan and truly keep track of it? I feel they should focus on the research and might lose track of their breeding plan, resulting in overproduction or missed timelines. So what does this a new breeding approach mean? Here we have the researchers communicating their requirements to the coordinators, who then come up with a plan and enter the necessary tasks in our line management system. At the same time, the purpose of each mouse is generally defined by the age of 4 to 6 weeks. Is this particular animal reserved for experiments? As a future breeder? Can we euthanize it because it has the wrong genotype or gender? Or if it's a surplus and it's a wild type, then we can offer it to our in-house research community. So what does this new setup mean for me working in the facility? Well, double checking is built into the system between the researcher and the coordinator, and then between the coordinator and the animal caretaker. Because looking at the top depiction, the researcher knows his requirements, and then he does the planning, and the animal caretaker basically just gets breeding tasks and has no idea what really is required. And if something in the planning isn't right, he cannot react and help to mend the mistake. And in the bottom, how we're set up now, the requirements, researchers know what they need, and then the coordination does the planning. And this entire thing ends up that the animal caretaker has a good understanding what is needed and what is important. And then if the coordination puts in well-defined tasks, it's hard to miss them. And gives also the animal caretaker the freedom to organize his weekly work in an efficient way. As the breeding tasks are set within a week and only if needed, and the coordination knows that that's important for us, only if it's really needed, we do it on a specific day. And that's then put into the corresponding request. Also, decision-making comes faster as the coordinators are always on top of their planning and can give quick feedback if needed. And this will also benefit the well-being of the mice. You won't have unnecessary keeping of mice with a finding or being single-housed for a prolonged time. And then also the 3 coordinators, in our case there are only 3, they can very well align to evade a clustering of tasks in a specific week. And I think this is virtually impossible if you have 30 people doing their separate breeding planning and then giving you the tasks. So who else benefits from these changes? I think it benefits the animals, your organization, and the researchers. I mean, fewer mice bred for the same number of mice used in research is good for everyone. Reducing cost and waste, that's good for your organization. Researchers have more time for the studies. I think that's a very important point. They can just give out their requests and then forget about it, and they can be sure to receive their animals in the right number, right genotype at the right time. And the coordination and the animal caretakers can easily take care of that. And faster decision-making benefits the well-being of your mice with findings or single-housed. And with this, we come to the summary.
I mean, basically to summarize, in the beginning, the managing of these colonies was really a challenge because we had a limited understanding of the researchers' needs, right? Then we had an initial optimization plan, and that resulted in a much better ordering ratio as shown, and in a 30% reduction of cage numbers. Second thing is the forecasting, and that's really allowing us now to plan ahead also and to understand what's coming our way. And really, if we had to summarize the whole talk, I would say that managing colonies turns out to be a very efficient way to manage space. With that, I would like to thank you for listening. I would also like to mention just very quickly that one of our coordinators is retiring and the role is posted on the Novartis homepage. So if we triggered your interest, feel free to take a look. And if you're interested, then feel free to apply. And with that, I'm going to stop. I'm going to stop sharing. And we are open for questions.
So thank you very much, Filippo and Daniel. I think my question, I think maybe Filippo already answered. So I don't know if this number that you gave are correct, but you mentioned there were around 30 researchers that asked, is this a real number or is this a made up number?
No, it varies, and it's but I talked to the coordination and they came up with 30 plus people they talk to and coordinate the breeding tests.
So then you mentioned, I think you mentioned, so with 3 people at the coordination team, it's enough to manage all this input from the researchers.
Yeah, it's a full-time. They're not in the facility. Like the animal caretakers do everything. They take biopsies and all hands-on work is done by the caretakers. And then the coordination just takes care of the planning. So it's a full-time job for 3 people.
And what kind of expertise what expertise of these people that are coordinating now?
In the current setting, the expertise is that they're all very experienced animal caretakers. Some of them also have taken project management courses. I myself have a background in animal engineering, so I know, for example, when you're doing CRISPR-Cas animal engineering, what to look out for in the first generations. So I'm here also to advise the coordination team. Having a good understanding of genetics is definitely helpful. We are currently also looking into options where somebody with a background in animal engineering might be beneficial because, as you know, in the CRISPR-Cas, there's a lot going on, and breeding initial stages of CRISPR-Cas models is very different from production breeding once this has been established. But basically, you need to understand the genetic side, and having really the experience, having been in a vivarium and understanding that the list of animals that you have in your database is something completely different than the racks full of cages and mice. That's also important because this is the biggest disconnect. You look at your animal list and you think, oh, everything is fine. And the animal caretaker stands in front of the rack and doesn't know what to do anymore.
Sorry, Omar, I need to step in a little bit. There were, I don't know, a lot of comments in the Q&A regarding the registration. I just want to spread out to everybody, that not everybody starts to ask again. we can see all the names, except I think one user is anonymous. So we can see all your names. You may not see your name by yourself because we have so many participants that you cannot see it as a regular Zoom meeting with all the people in front of you, and to see your own name. But don't worry, we can see all your names and you all are registered. Just in one person, I was answering directly that we should register him by him sending his name in. That's the only one. Okay, so there are some questions in the Q&A section, Homari, if you may have a look on them.
Yes. Yeah, so we can start. Anastasia is asking, so doing there's a difference in breeding throughout the season. It seems to be that during fall, winter, for example, there's some particular issues. I don't know if it's a reduction in breeding. So how do you, is there something in your facility that you need to control for these seasonal changes, or inside the facility, do you see a change?
So you mean breeding performance?
Here is particularly, this says that there are many reports of stress response changes during fall season, and we have seen changes in glucagon secretion in our lines. This is particular question about stress, but maybe.
So, I mean, we have mostly an overview over what's going on in the breeding, right? So Filippo and myself are not directly involved in running experiments. So on that side, I cannot comment. I mean, regarding the breeding, there is discussions whether the weather and everything and summer, winter can have an influence. It's not something we have analyzed in a lot of detail. What I also have to say is that we have had to move our breeding facility over the past 10 years, I think 3 to 4 times, right? So even if we had data, it's different facilities, it's different buildings. And so I'm really unable to answer that question. Generally speaking, what we do see more is the ups and downs regarding the breeding requests coming from the researcher, but I think that was not the question.
Okay, no. Then the second question, but Manya, is about why do you do breeding internally and not just buy them? I guess that's the summary of the
because
question.
they're not available. They're not available, right? So if you look, for example, from Jackson, you can get many lines, but certainly not everything you want, because there are well-established lines there that others have already used. For us, we generate a lot of lines internally, and then we analyze them, and we may share them later on. So they are just not available out there. And very often you have situations that a well-established line and model has been published by, say, a university, and our researcher want to work with that exact line. And then, I mean, it's not possible to order experimental cohorts, for example, from a university in the US. There's transport, there's hygiene discussions. So for these reasons, we set up many, many lines internally because we simply cannot purchase them from externally.
Then for Filippo, I mean, there's this idea that, well, what's your opinion on keeping surplus animals in the facility until they die on natural causes instead of euthanizing them?
Well, the surplus mice we keep as companion animals for future breeders that they're not single-housed. But keeping them till they die, I mean, we have a lot of constrained lines too, that the constraint shows up with 2 months, 3 months, 4 months. And by not letting them sit so long, we don't run into those constraints. And then they are impaired and they're not feeling good. So,
and maybe to add to that, right? So the question is always, when do you stop? I mean, imagine you have a cage with 2 males or with 3 males, and you want to let them sit. They have an established order between them. Then one of them maybe gets a wound or something, and you have to euthanize it for humane reasons. Then you have 2 sitting there, and they reestablish the order. They start fighting, which is also something you don't want. And then ultimately there will be single-housed animals left, right? So you really need to weigh all these factors. And for us, I mean, honestly speaking, it's not really a discussion we have had in the past. But I think there is so many variables that you need to keep in mind, right? What Filippo mentioned, what I mentioned. Are you then going to keep a single-housed mouse until it naturally dies? Is that better than killing it at some point? And I mean, for me, it's also really an ethical discussion where you need to weigh all these different factors.
Yes. Do we have time for another question? Oops, they're moving. Something that will. So one quick question was, I mean, there's the availability of this breeding software that Philippe Bugnon also developed. Do you use a breeding software? How do you do the calculations actually to determine how do you calculate the breeding?
So we do use and we do test these softwares that become available. So there's not only this one out there, but recently, or like a year or two ago at the ISTT, another one has been presented. You really have to differentiate here between production breeding with a well-established line where you have, let's say, 50 homozygous animals sitting there and you need to produce batches, and between establishing a line. So we look into it, but it's usually not something we use on a routine basis, because for establishing our lines, that's the slides in the beginning. You really have few animals and you depend on the correct genotypes. And then we obviously know what we have to start with. We do a calculation, but then the next step, you really need to see what comes out of this. You cannot really plan over 3 or 4 generations when you're starting crossbreeding with limited numbers of animals. And then usually once we have the line established, we have a good understanding of the breeding performance. So we know that we will get on average 5 or 6 pups per litter. And then that's pretty much straightforward.
Okay, I think that's some more question, but I think we need to move to the next talk. So thank you very much, Filippo and Daniel.
Thank you for your time.
Yeah.